Hey, hi student. Video number 3, Mendel's law of inheritance, 2 gene, dihybrid cross. Okay. So before going to the concept, here we are going to see about back cross, test cross, dihybrid and the concept of dominance. So first one is back cross. Okay. So back cross is nothing but crossing between a hybrid and any of its parent. So let's see how it is done. Okay, so yesterday we have seen a few concepts, right? Like chromosome leda gene irukku. So we have seen that, right? So this is the actual example. So this first image which shows the karyotyping. This is how a chromosome is visualized. Okay, la. Second image pathina, this is the database which is present in the genome database. So adala chromosomes la we have these black color bands, right? These are nothing but the gene which is mapped into it. Okay, these are all the genes. So these are all the gene pattern which is mapped into it, and this is chromosome number 1. So we start with the concept of back cross. So back cross abdingumbodhu we are going to take the hybrid. Okay, hybrid, and you're going to cross it with either a true breeding homozygous dominant or a true breeding homozygous recessive. So rendume capital T, isn't it? So adinala we call that as a homozygous dominant. Okay, and this one is homozygous recessive. Okay, so idoda results are based panni he made a protocol. Okay, so let's see how this table is done. So we very well know nammaloda hybrid gamete capital T small t. So I'm going to cross it with true breeding dominant plant. Okay, so capital T and capital T. So what are the results? Capital T, capital T, capital T small t, capital T, capital T, capital T small t. What are the results? I'm going to have, the results are phenotypic abdi namba paathona, phenotype. Okay, phenotype physical paathona, all the plants are tall. They are tall. Okay, so all the plants are going to be tall. All the plants are tall. And if I am going to cross the same thing in the hybrid if I am going to cross it with a recessive plant or recessive parent, okay, let's see how it is. So the hybrid capital T small t, and I am going to cross it with a homozygous recessive. So I am going to cross it with homozygous recessive parent. So small t, small t. So capital T, small t, small t, small t, capital T, small t, small t, small t. So these are the results. இப்போ இதோட phenotype ratio எப்படி இருக்கும்? Phenotype tall plant, short plant, and this is also short plant. So 50% of tall plant and 50% on the short plant. And what will be the genotypic ratio? Genotype is also same. Okay, so I have 2 capital T small t is to 2 small t small t. So cancelling the 2, I get 1 is to 1. 1 is to 1 ratio. So these are the 2 things which you should remember under the back cross. So what if the hybrid is crossed with a dominant parent or what happens if the hybrid is crossed with a recessive parent. Okay, so in the results vachittu Mendel was able to understand the genotype of the hybrid. Now there is specifically in the genotype understand pandradhukku he brought up with a new concept, that is going to be the test cross. Okay, test cross. So this is how the test cross is done. So it has been given in the NCERT book. The concept here is we have Mendel want to know what is the genotype of this violet flower. There are two probability. ஒன்று ரெண்டுமே capital W இருக்கலாம், or it can be capital W and small w. He crossed that with the true breeding recessive parent, homozygous recessive. Underline homozygous recessive. Okay, so homozygous recessive parent kuda he flower kuda he crossed it. Let's see what is the results. I have a violet flower, and there are two probability of its genotype. Either it can be capital W or capital W and a small w, right? So what he did for test cross, he crossed option number 1 and option number 2, he crossed with the recessive parent. So first one papo, option number 1, capital W, it is crossed with the recessive W or homozygous recessive parent. Okay, so capital W, small w, capital W, small w, capital W, small w, capital W, small w. So what will be the color of this? Violet, again, violet, and this is also violet. Why so? It is because all these capital W will not allow the recessive one to express, so they mask it. So the results going to be, just see here, all flowers are violet. Okay, unknown flower. So this gives a conclusion, namakita irukka unknown flower vandu homozygous dominant. Okay, this is the conclusion. Now the second option we try. Okay, so namakita irukkiradhu maybe in the genotype ah irundadana, so Mendel cross பண்ணும்போது, see here, capital W and a small w, option number 2. Okay, and I am going to take the recessive parent which codes for the white color flower. So see here, capital W, small w, small w, capital and small, small and small. So what you can see here, first one vandu violet, second rendume small w, so it's a white color flower. Once again violet color flower, and this is white color flower. Okay, so what it implies? It implies that 50% will be a violet flower and 50% will be the white flower. 50% will be the white flower. So Mendel come to a conclusion that enkita irukra the unknown flower, the unknown genotype irukra flower vandu, it should be a hybrid variety, hybrid. Okay, hybrid genotype of capital W and a small w, hybrid variety of capital W and a small w. So this is how Mendel try to understand about the genotypes. Okay, so are you clear with it? Law of segregation, which states that, see here, during gamete formation, okay, gamete formation, the factors or the alleles, so these are the alleles, capital T, small t, so each chromosome la oru gene pair irukum illaya, they are the alleles which segregate from their parent, okay, So they segregate from their parent. Gamete formation appa they segregate separately. Illaya? So homozygous parent, if the parent is homozygous, okay, and the gametes also similarly they produce a similar gametes. If the parent is heterozygous, they produce a dissimilar gametes. Avladha. This is the concept of law of segregation. So one allele with equal proportion, each gamete will get one allele. Okay, the next concept is dihybrid cross. So in dihybrid cross, there are two concepts which is taken here. Two contrasting characters are taken here. Okay, what are the two contrasting character? Seed oda color and seed oda shape. Okay, they are the two characters which has been taken here. Let's see. So in a dihybrid cross, we take two characters. So it's going to be the seed color and seed oda shape. Okay, so these are the characters. So what is going to be the dominant and recessive trait? Okay, so look at here. So we have a dominant trait and recessive trait. Okay, so dominant trait, seed color வந்து dominant color எது? It's going to be yellow, and recessive trait வந்து green. Okay, seed shape வந்து round, dominant, and recessive வந்து wrinkled. So yellow ku how I am going to represent as capital Y, capital Y. Green ku small y. Round na capital R, capital R. Wrinkled na small r, small r. So idoda concept yesterday we have read about the chromosome and the pattern, right? So adula understand that. So this is one chromosome pair. So in the chromosome pair la, this one codes for seed color and this one codes for seed shape. அதே மாதிரி தான் இங்கேயும். Okay, right. So homozygous round yellow அப்படின்னும் போது capital R, capital R, capital Y, capital Y. Wrinkled green, small r and small y. Okay, so let's see what are the gametes it can produce. See here, capital R, capital Y crossed with small r, small y. Okay, right. So this is the parent generation. So இதோடைய gametes என்னவா இருக்கும்? It is going to be capital R, capital Y. Okay, and இதோடைய gamete வந்து small r, small y. Okay, so your F1 generation you're going to have, see here, capital R. So I'll write here capital R, small r, capital Y and small y. Why I should write like this? Because capital A number small r முதல்ல எழுதக்கூடாதுன்னா, it is always a rule that you should write the dominant one first followed by the recessive one. Okay, so this is a hybrid and this is the F1 generation. And இது என்னது? This is the gametes. Okay, right. So what is the next step I do? I'm going to do a selfing. So gametes enna possibility irukku? Capital R, capital Y, idu oru gamete, and once again capital R and small y, adu oru gamete. Okay, adhe madri small r and capital Y, idu oru gamete, and small r and small y. Okay, small r and small y, idu oru gamete. So idu parent 1 ku. Adhe madri parent 2 ku we will be writing the gametes. Clear? Are you clear with this? So in the step vandadukku apparama it is very simple. The next thing, the Punnett square, is very simple. Let's see the Punnett square, how it is done here. So the first gamete vandu we are going to write capital R and capital Y. Okay, capital R and small y, small r, capital Y, and small r, small y. Adhe madri second parent odaya gametes vandu we are going to represent here. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So combinations paranga. So we are going to write capital R here, and capital Y, capital Y. So what is the phenotype of the rendu R irukanaala it is a round yellow, round yellow. Okay, right. Next up, capital R, capital R, capital Y, small y. So round, once again y irukku, so yellow, round yellow. Okay, now capital R, small r, capital Y and capital Y. Idu round yellow da, round yellow. Now capital R, small r, okay, capital Y, small y, round yellow. Next, capital R, capital R, capital Y, small y, round yellow. Capital R, small y, small y. Okay, R ku round எழுதியாச்சு, and if it is going to be small y, which codes for green color, round green. Okay, இதை mark பண்ணிப்போ. This is one hybrid. Next, capital R, small r, capital Y and small y, round yellow. Next, capital R, small r, small y, small y. Once again, round green. Okay, right. Now you have solved the Punnett square. Just you will be telling me once again the proportion. Okay, so these are the one. We'll just take it. Round yellow. So we write round yellow. Okay, how many round yellow we have? So totally 9 round yellow. Alright, and idoda genotype variations la parunga. Capital R, capital R, capital Y, capital Y. Ipdi irundhalu round yellow. Capital R, capital R, capital Y and small y. Ipdi irundhalu it is round yellow. Okay, capital R, small r, okay, and capital Y, small y. Ipdi irundhalum it is a round yellow. These are the different types of, what you'll say, genotypes. Types: wrinkled yellow, this one, wrinkled yellow 2, and wrinkled yellow 3. So wrinkled abdinale how many? Okay, totally 3. So we have small r, capital Y, this can be a genotype, and small r, capital Y and small y. These are the two types of genotype which shows wrinkled yellow. Okay, so we are going to see the round green here. So round green, let's see how many are there. Number 1, round green 2, and round green 3. So what are the genotypes you can observe here? It is going to be capital R and capital R. Okay, round. Green ku small y. So genotype 1, capital R, small y. Okay, these are the two genotype for round green. Alright, okay, and the last one is going to be the wrinkled green. So we have only one wrinkled green here. So both are recessive genotype, small r, small y. That's it. So 9:3:1 ratio. Okay. One actually I missed it. It is the capital R, capital Y for the round yellow. So totally we will have 9 different genotypes. Okay. So 1, 2, 3, 4, 5, 6, 7, 8 and 9. 9 different genotypes we have. See, this is the ratio just now we have seen. And this is going to be the genotypic ratio. So that's it. We are done with Mendel's law of inheritance with a dihybrid cross. Hope it will be useful for you. Thank you.